Hey everyone, today we have lab exam 2 exercise over lab experiments 10, 11, 12, 13, 14, 15, and 16. So for the best result of this exercise, please repeat lab experiments 11, 12, 13, 14, 16, and 16, as many as possible before taking this exercise. Prepare some pieces of paper and pen or pencil to write your answer. There will be 2 or 3 seconds after each question to write your answer on paper before the answer key is given and explained. Remember, you can always pause the video if you need more time. After finish the exercise, check your answers, how many are correct and how many are incorrect. Repeat the process until you get 100% correct answer. Make sure that the 100% correct answers are based on knowledge, not from guessing. It is very likely that you will get a very good score on your lab exam number 2 if you follow this instruction correctly. Okay, we start with question number 1. Which structure is not trimmed away in eye dissection? So during the eye dissections, which part of the eye will not be trimmed? A, B, C, or D? Okay, to answer that, look at this eyeball. Okay, the eye that will be dissected. So it has a lot of things over here that has to be removed actually. Like for example, this muscle over here, it has to be trimmed in order to be able to dissect the eye. The fat tissue has also should be trimmed. And if there is still eyelids over here, then this eyelid has to be removed also. The only one that has to stay is this part over here, which is the optic nerve. The optic nerve should stay in the eye when you perform the eye dissection. So the answer will be the optic nerve. Number 2, the optic nerve, which is cranial nerve number 2, carries the spatial sense of A, B, C, or D. Should be easy answer because our lab experiment is about eye, so it's going to be vision. Where do you perform a corneal cut in an eye dissection? A, B, C, or D? Okay, look at the eye that we have. So dissection has to be performed through this tissue called the sclera, round dissection. The sclera just behind this cornea. Okay, so the area through the sclera just posterior to the cornea. So we cut it round, okay, until it's divided into 2 sections, the front sections of the eyes and the back section of the eyes. Visual acuity is measured using A, B, C, or D. So you did this experiment using this chart to know whether your vision is normal 20/20 or not. So which one we use? So we use chart like this, and this chart is called the Snellen eye chart. Okay, so we see the Snellen eye chart. They have different size of letter. Now, number 5, if a person sees the Snellen chart just like this from 20 feet, which following condition could be excluded? So if you're able to see this chart, all the letters on the chart from 20 feet, especially this 20/20 over here, okay, to 20 font size from, okay, from 20 feet. So this person is considered as having a normal eyes. It is called a 20/20 vision, which is normal eyes. So if you're able to see this, then you are in normal eyes, okay, which is here. Okay, now which one can be excluded from this? So if you're unable to see this, it means that you have to go closer to see this chart. So when you go closer, it means that you have condition is called the myopia, okay, which is nearsightedness. You're unable to see. You're able to see close object, but unable to see far object. So you are having this condition. You exclude this myopia conditions if you're able to see this Snellen chart from 20 feet. Stigmatism is assessed using A, B, C, or D. Okay, so the chart for stigmatisms is look like this. So if you see this one, so if you have stigmatism, this 3 line over here, okay, so some of this 3 line will only look like one thick line. Okay, so this is indication of stigmatism, and this chart is called a stigmatism chart. Astigmatism is a result from a defect in the A, B, C, or D. So which causes stigmatism? So this is mostly caused by the cornea, irregular shape of cornea. So this should be the answer of here, or sometimes lens can also cause stigmatism. So cornea or lens. Stigmatism is mostly caused by a problem with the A, B, C, or D. So I just mentioned before, it is the shape of cornea that causing the stigmatism. Normal people will have a soft cornea, okay, but people with stigmatism, maybe the cornea is a little bit rough, okay, it's not soft. So soft, this is kind of rough, okay, or irregular shape of cornea. Okay, so for normal peoples with the soft shape of cornea, they will be able to see this letter A clearly, but if they have stigmatism, that letter will be blurry or sometimes become thicker, because it's going to be kind of a double. The one that double become look like one thick line or letter. unable to read a clock when it is 11 o'clock, but reading is fine with other times or numbers like maybe 2, 3, 4, is a sign of the A, B, C, or D. Okay, if you see 11, it means that there are two lines that is close together. So if you're unable to see these two lines close together, maybe you only see one line, that's a sign of stigmatism. Okay, so the side over here. Look at this. Number 11 over here. If someone with stigmatism, this number 11 maybe just look like a thick line like this. Okay, look like number 1, but thicker. So that's stigmatism. Blind spot is defined as the area A, B, C, or D. Okay, so the area that have the photoreceptors is called retina. There are some retina area that actually do not have photoreceptor, like rods and cones. So if you see this picture over here, okay, this retina, which is the farther back part of the eyeball, is consists of this photoreceptor, rod and cones. Rod is for black and white vision. Cones is for color vision. So there are some area in the retina that do not have this photoreceptor, and this area is called the blind spot. Okay, is over here. Okay, so this blind spot is the area where the nerve fibers leave the eyes and form the optic nerve over here. So this should be the answer. Okay, retina filled with the rod will be wrong because this is photoreceptor. This is also photoreceptor. It's a wrong answer. Lack electricity is not correct. It should be lack of photoreceptor. That's the correct answer. So for this one, D will be the best choice. 11, the blind spot is located at the blank of the eye. Okay, we just mentioned that it is located in the area is called the optic disc. Okay, so optic disc is where the place no photoreceptor, and it is known as a blind spot. The blind spot or optic disc is in the blank visual field of the eye. A, B, C, or D. Okay, look at this picture over here. So when the light enters into our eyeballs, it's going to the center area over here. Okay? The center area is called the central fovea. Okay? Central fovea. This is where color photoreceptor, the cones, are located. So if the object goes to our central fovea, then you can see the object with color. Okay. Other area than central fovea, like this area or this area, okay, is called the lateral. Okay, so lateral vision, including the blind spot over here, is located on the lateral area. So that should be the answer for that question. The blind spot does not have any blank and therefore produce no visual image. So which one is that? Nerve fibers, rods, cones, rod and cones. So the answer should be no, does not have any photoreceptor. Photoreceptor. Okay, and There are two types of photoreceptor. For black and white, is called rod photoreceptor. For color, is called cones photoreceptor. Okay, so it does not have rod and cones. So D should be the answer. What are rod cells? A, B, C, or D? Yeah, we just mentioned that the rod cell is a photoreceptor that detect black and white. Okay, so this photoreceptor detect the color. So this should be incorrect. This photoreceptors that detect the quality and quantity of the light is not really correct, because with quality, quantity lights, you will see the color too. They are photoreceptors that detect the bright of the light but no shade. They are photoreceptors that detect the amount of light but no color. So remember, this is the key answer over here, no color, as the functions of the rod photoreceptor to see object, black and white object. Normal type of visions of 2020 is called A, myopia, hyperopia, emmetropia, or nyctalopia. So this is terminology for normal vision is called emmetropia. Okay, so this is normal visions when the object actually go to the central fovea, okay, to the retina. If it is myopia, which is nearsightedness, the object actually in the front of the retina. It means that you have to go closer in order to make the object go to our retina. So this is nearsightedness. Hyperopia, farsightedness, this is when the object actually go behind the retina. Okay, therefore you have to go back. You have to move back to in order to see the clear object. Okay, this is hyperopia. Stigmatism, this is caused by the irregularity of the cornea. So you're gonna see three lines as one big line or thick line. So that's emmetropia. Okay, nearsightedness vision is called A, B, C, or D. So you're able to see. Okay, near sight means you're able to see closer object. Okay, but you cannot see farther objects. So this is nearsightedness. And this is terminology for myopia. So myopia, if someone have a difficulty to see farther object. So they usually have to wear glasses in order to make correction of their vision. Farsightedness vision is called, okay, of course it is not myopia because this is nearsightedness. Emmetropia is normal vision. Okay, the answer will be hyperopia. This is for farsightedness. Okay. Next one. The difficulty seeing in low light condition is called, okay, of course it is not normal eyes, it is not myopia, hyperopia, we already know what is that. So the answer will be nyctalopia, and usually it is caused by lack of vitamin A, okay, on their diet. So these people will be very difficult to see during night time. condition of older people, when the lens decrease its ability to accommodate near vision, is called A, B, C, or D. It is not normals, it is not nearsightedness, it is not farsightedness. It is called a presbyopia, and it is caused by the lens. Okay, the elasticity of lens is decreasing. So this person usually unable to see close objects. So they have to, like reading the newspaper, they're gonna be, the near vision will be farther. The near points of accommodations, A, B, C, or D, whether remains unchanged within the eye, the age, decrease with age, increase, or assess using the Snellen eye chart. So this one is not correct. Okay, so accommodations, whether remains, it is not correct also because it will change. So the near point means the closest object that you can see with focus. When we are older, then our near point will be increased. Okay, so we cannot see the very close object. We have to move the object a little bit away from our eyes. That's when the old people have problem reading the newspaper. Okay, so the problem is when we have accommodations, when we see the near objects, our lens is become bulging. Okay. When we see far object, the lens is flattened. Okay, so when we are older, we lost this ability to make the lens become bulging. So therefore, we cannot see the near object. So it's gonna be farther, okay? Because our lens is actually not really bulging, it's kind of still flat. Then the object that we see will be farther. So our near points will be here. Okay, so in average, normal near point for children is around maybe 5 to 6 inches. So for us, maybe increasing above 10 to 20 inches, maybe. Okay, so that's the the increase of near points within the increasing of the age. This is caused because the lens elasticity is decreasing. Does the elasticity of the lens increase, decrease, or remains the same within age? A, B, C, or D? So the elasticity will be decrease. Okay, so our lens is not elastic anymore. Okay, so that should be the answer. So we are unable to perform these accommodations. Okay. Color blindness involves inherited defect of rod cones, blue, red, green, red and green. Okay, so in order to answer this, look at this picture over here. So the photoreceptor that used for seeing colors is called cones. Okay, and we have 3 types of cones: blue, red, and green. And with these combinations, then we will be able to see different spectrum of colors. Just like color printer that we have, usually only have 3 cartridge color. Okay? So blue, red, and green. Now, people usually do not have problem with blue. So blue is always present. Okay? But some people actually, they do not have or lack of red or green or both. Okay? So this is the one that causing the color blindness. So usually color blindness related to color red and green. So the answer should be C, T, which is red and green. Okay, if you see this one, the rod, which is for black and white, okay, and then there's a blue, red, and green cones. So rod is actually about 91 million rods in our eyes, compared to cone is only 4.5 million cone photoreceptor in our eyes. So rods will be spread all over the retina, while the cones usually located in the central fovea. So this is the time when we see a color object. People with a decreased sensitivity to green light are said to have this type of color blindness. A, B, C, or D. Remember, the color blindness is usually related to green, okay, green and red pigment. Okay, so usually some people do not have green, or maybe it can be, maybe 25% lack of green, 50%, 75%, or maybe 100% lack of this pigment. Okay, so if it is related to greens, lack of green pigment, this condition is called the deuteranopia. Okay, deuteranopia. So this is for terminology for green color blindness. Now, how about for the red color blindness? Okay, so this is red. The terminology is called the protanopia. So protanopia is for terminology for people with the red color blindness. The functional unit of the muscle tissue are so we are moving from eyes, from visions to muscle lab experiment now. Okay, the unit of muscle tissue is called, of course, it is a neuron, because neuron is for nerve tissue. Okay, nephron, this is for kidney. Okay, neuroglia is also for nerve tissue. Okay, so the correct answer will be sarcomere. So sarcomere is the functional unit of muscle that actually contract. So if it is the whole muscle, if we look closer, there is consists of many fascicles, and each fascicle consists of muscle fiber. Inside the muscle fiber, then you see this stripe of thick and thin filament. And this unit is called the sarcomere. So when the sarcomere contract, become closer, then the whole muscle over here also will be contracted. So the name of the unit is called the sarcomeres. A muscle cell is also called as A, B, C, or D. Okay, so muscle cell, one muscle cell over here, okay, is called muscle fiber, muscle cell, or myocyte. Okay, so it could be myofiber, myocyte, muscle cell. So the answer will be A and B are correct, myofiber or myocyte. The cell membranes of a muscle cell is called A, B, C, or D. Okay, of course, it's not sarcomere, because sarcomere is the unit, contractile unit inside the muscle cell. Sarcoplasm, the name is telling you this is kind of cytoplasms. Okay, this will be not the answer. Sarcoplasmic reticulum, it is look like kind of ER endoplasmic reticulum. So this is not the answer. So the answer will be the sarcolemma. So sarcolemma is the same as a cell membrane. For regular cell, it's called the cell membrane, but for muscle cell, it is called sarcolemma. So this membrane over here is called the sarcolemma. What's the function of sarcoplasmic reticulum? A, B, C, or D? So we see this picture. So it has sarcolemma. It has these myofibrils that there will be sarcomeres over here. Okay, and it has this structure which is called the SR, okay, sarcoplasmic reticulum. And the function of the sarcoplasmic reticulum is to store what? Glucose? No. To store glucose is called glycogen. Okay, that's the storage for glucose. Storage for a release of oxygen gases. It's not. The structure that carry oxygen is called the myoglobin. Okay. It is not sarcoplasmic reticulum. Energy production, of course, it is not, because energy production is mitochondria, right? Then the answer, the correct answer will be storage for calcium ions. That's the function of SR. The thick filament of sarcomere is composed of protein. Is the A, B, C, or D? Okay, look at this picture right here. There are two main filaments for sarcomere. The thick one is in the middle over here. It's called myosin. And the thin one is on the side. And this is the one that will be sliding to the middle when muscle contracting. And the thin element is called the actin. So thin actin. Okay? So just remember that. So the question is the thick filament. Okay, so the thick filament will be the myosin. The thin filament of a sarcomere is composed of a protein. Is the, we already mentioned that, thin. Okay? So this should be the answer. Actin is the thin filament. The interaction between actin and myofilaments in the sarcomeres are responsible for A, B, C, or D. Okay, of course, C, D will be incorrect. So the question is between these two. And remember, the sliding of actin and myosin will give the muscle contraction. Okay, if you see over here, this is when the muscle is relaxed. So there is no interaction between actin and myosin. So therefore, this one is incorrect, right? And then when there is interaction, which is the actin sliding to the middle, then the sarcomere becomes shorter. Okay, this is when the muscle is contracted. Okay, so contraction is actually when the muscle becomes shorter. And this is happening when there is interaction between actin and this myosin. So this is the correct answer. According to the sliding filament models of muscle contractions, the myofilaments sliding each other results in overlapping of myosin. And which one? Again and again will be actin. So actin, myosin, these are the two filaments that slide each other during muscle contraction. Okay, so this is not correct answer. Okay, so it should be actin. that, the correct answer is overlapping of myosin and actin. The regulatory protein of sarcomere that bind the calcium release for the SR is the A, B, C, or D. Of course, it will not be actin and myosin because this is the primary protein for contraction. So the answer will be this one, okay? Regulatory protein, whether tropomyosin or troponin. Look at this picture over here. You see this one? This is myosin, okay, the thick filament. And you see this one is actin. So actin actually has two other regulatory protein on it, okay, which is the tropomyosin. It's a long protein that cover the binding site of this myosin head. Okay, so in order to contract, this myosin head has to bind to the binding site on the actin. During relaxation, this binding site covered by tropomyosin. So tropomyosin is not the answer for this one, okay? Because it's a bind. So this is the one that covers. The one that binding the calcium will be the troponin. And this calcium is released from the sarcoplasmic reticulum. So the correct answer will be troponin. The regulatory protein of sarcomere that cover the myosin binding site on the actin is called, okay, this is tropomyosin. So we already mentioned that this tropomyosin is a long type of protein that cover the binding site during relaxation. Okay. So in order to contract, this binding site has to be available for the myosin head. The contraction process in a skeletal muscle is initiated by a release of blank ions from sarcoplasmic reticulum. This will be easy answer because we already mentioned many times that SR function is for storage and release of calcium. So that should be the answer. So this release of calcium then will bind to the troponin. And when it is bind to the troponin, the binding site will be available. Then the myosin will be able to slide or to pull the actin for contraction. As their concentration in the sarcoplasm increase, calcium ions bind to blank, changing its shape and liberating tropomyosin from actin binding site. So bind to what? Calcium, bind to troponin. Okay, that's the regulatory protein that bind the calcium. Once calcium has been released and enable actin-myosin crossbreed to form, blank is needed for muscle contraction. So what is next needed in order to muscle to be contracted? So we already have the calcium. Calcium already bind to the troponin. The binding site is available for the myosin head. The myosin head bind to it to make what we call the crossbreed. The next thing that we need is energy. Okay, energy for contraction, which is provided by this ATP. So ATP have to be provided or available in order to contraction to be started. Okay, so ATP will be there. So if you see over here, when the calcium available, the calcium will bind to the troponin, and then it will remove the tropomyosin from binding site. The myosin head will be able to bind to actin, and this binding is called a cross bridge. Okay, and then after that, with the ATP, the myosin head will pull the actin. This process is called the power stroke. Okay, and then in order to detach this actin, in order to be relaxed, then another ATP has to come bind to this myosin head. So this is detachment. Okay, so the myosin head will detach the actin and then will release this, causing this relaxation. So relax, this is contract over here. When the actin is pulled by the myosin head. That's the cycle of muscle contraction. Which of the following lists the part of a reflex are in correct sequence? So in a correct sequence. A, B, C, or D. Okay, so our nervous system work. Okay, start with, of course, when we receive the stimulus. Part of the nervous system that receives the stimulus is called receptor. So that should be the first one over here. Okay? And then that receptor will be carried by the sensory neurons. So the next one will be sensory neuron. Sensory neuron carry the information to neurons in our brain and spinal cord. That neuron that process information in the brain and spinal cord is called the interneuron. And then brains will produce the response. The response should be carried by the motor neuron into the effector. And effector is the one that performs action. Example is muscle. So muscle is example of effector organ that actually perform action based on order from the brain over here. Okay, so that's the correct sequence. From receptor, carried by sensory neuron, processed by the interneurons in the central nervous system, brain and spinal cord, and then produce the response carried by the motor neuron, and the action will be performed by the effector. Okay, so receptor, sensory neuron, interneuron, motor neuron, and then effector. This is only one way. Okay, so going this way and end with the action of the effector. What neurotransmitters is released at the neuromuscular junction when an action potential arrives at the motor neuron terminal? Okay, so our muscle contraction is actually ordered by our nervous system. And again, muscle is an effector. The one that ordered that, carry the order from the brain, is called motor neuron. Okay, so this motor neuron will release a neurotransmitter. The one that releases to the muscle is called the acetylcholine. So this is the neurotransmitter is carried by this motor neuron that will be released in the area between this motor neurons and the muscle tissue over here. So this is the acetylcholine. Okay, if you see this motor neuron carry the informations or respond to the many muscle cells. It's not only one muscle cell. One motor neuron can innervate or bring response to many muscle cells. Okay, so this junction is called the NMJ, neuromuscular junction. And the one the neurotransmitter release to this junction is called the acetylcholine. All the fibers in one muscle that a simple motor neuron innervates make up A, B, C, or D. Okay, remember, because this is carried by the motor neuron, Therefore, this unit is called a motor unit. So one motor neuron with many muscle that it innervates, it's called one or a motor unit. A motor unit is a composed of A, B, C. Okay, remember, composed of only one motor neuron. Okay, now it innervates several or all the muscle fiber that this motor neuron innervates. It should be all. Okay? It's not only several. All muscle fiber that innervate by one motor neuron is called one or a motor unit. Okay, so only one motor neuron with all of the muscle fiber that it innervates. That's a motor unit. To make a muscle contract more strongly, the nervous system can activate more motor units. So it's not only one motor unit will be activated, like we have motor neuron, then innervate several muscle tissue over here. Okay, and there will be maybe other neuron that innervate other muscle tissue. So this is one motor unit number one. This is motor unit number two. Okay, so in order to make a stronger contractions and meaningful contractions, the nervous system able to recruit more motor unit to order the contraction. So this process is called a motor unit recruitment. Okay, you see this one, there are two motor units that actually recruited in order to make muscle contraction in this group of muscle. Motor unit recruitment is the blank of motor units to blank the overall contraction strength of a muscle. So what is this? What is motor unit recruitment? It is the addition or subtraction. Of course, this is going to be addition because more than one motor unit will be added into this contraction. To do what? Increase the strength or decrease? Of course, it is to increase the strength of the muscle. So this one should be the answer. An EMG, electromyogram, measures electrical activity in where? Any type of muscle? No, we cannot measure, we cannot use EMG for any type of muscle. For, like, for example, cardio muscles, we will not use this one. Or for smooth muscle, we will also not use this EMG. So this EMG is only for use only for skeletal muscle measurements. Okay, the contraction of skeletal muscle, the strength or the weakness of the skeletal muscle contraction. So if you perform this lab experiment, you should be able to answer this question easily. A recording from an electromyograph or EMG is actually a recording of what? This is motor neurons? No, it's not measuring the motor neuron. It is measuring the muscle, right? So it is not measuring the skin, so this one should be the correct answer. Muscle electricity. A recording from an electromyogram or EMG is a recording of what? Motor neuron action potential is not really. Skin is not. This is actually how many motor units activated to the muscle contraction. Okay, so there is a muscle contraction over here related to this motor unit activation. So this should be the answer. When sodium flows through the open voltage gated channel, the membranes will A, B, C, or D. So just remember, I said this is a cell. Okay? The cell has, outside the cell, there are a lot of sodium ion. Okay? Inside the cell, there will be a lot of potassium ion. Both of them are positive. Okay? When the sodium ion channel gated channel open, then sodium will be entering into the cell, and the cell become positive. And when the cell become more positive, this called the depolarization. Okay, so depolarization. So that should be the answer. So when the sodium ion open, the electricity in the cell increase, so positive increase, and this process over here is called the depolarization. Now look at this one over here. The electricity decrease, which is the positive charge decrease, is because of when these potassium ions get it channel open, the potassium will go out from the cell. Remember, potassium is positive. So when positive leave the cell become more negative or less positive. It is going down. And this process over here is called the repolarization. Okay. So depolarization when it is increased, repolarization when it is decreased. Depolarization is caused by sodium that enter into the cell. Repolarization is caused by potassium that leaving the cell. Okay, so 47 should be depolarization or depolarized. Now, when potassium, okay, ion flow through open the voltage channel, remember there are more potassium inside the cell. When they are leaving the cell, then what happened with the cell? It's become more negative, and this is called the repolarization, more negative charge. Okay, so in the EMG, in the graph will be going down like this. Okay, this is depolarization. I mean, this is repolarization, sorry. Okay, depolarization increase, repolarization decrease. Each motor unit action potential, or MUAP, recorded in the electromyogram or EMG, correspond to the A, B, or C. So this is correlated to the sum of the action potential from all the muscle fiber. Action potential from all the muscle fibers. Yes. It's not only single muscle. Okay? It's not individual. So this is all muscle fibers will be recorded. Now look at this one. Which of the four recorded motor unit action potential or MUAP correspond to the strongest muscle contraction? Which one is going to be? This would be easy to guess because this is a stronger graph that show that is a stronger or strongest muscle contraction. And the graph number 4 over here. So which of the four recorded motor unit action potential of MUAP correspond to the smallest number of muscle fiber contracting? Okay, of course, it's going to be this one. And that's the weakest contraction. Muscle are connected to the bone by A, B, C, or D. Okay, so we have to remove actin and myosin because this is not connection. This is inside the muscle, so it's not correct. So the choice between ligaments and tendon. Ligament is connective tissue that connect bone to bone. Okay, so bone to bone is called ligament. Tendon is the connective tissue that connect muscle to bone. Okay, that's the terminology, that's the name. So the correct answer will be tendon. Look at this one over here. Okay, bone and then muscle. There is connection, and it's called tendon. The highlighted muscle in this cadaver is the A, B, C, or D. So you have to memorize the name of muscles and location. So this one is located in the front part of the anterior part of the upper arm. Okay. So this one is bicep. Now how about this one? This is located on the back part or posterior part of our upper arm. It's going to be opposite of the bicep will be the tricep muscle. In the shoulder elbow movement, so this is the lab that the experiment that you perform. And in the shoulder elbow movement lab, this muscle is prime mover for forearm flexions at the elbow joint. Okay, so if we have elbow joints like this, okay, this is the one that move our hands, flex. Okay, our hands in the elbow joint here. So which muscle is that? So that muscle that flex elbow joint will be muscles in here, in the front part. Because when it is contract, it will bring our arms or flex our arms. So this muscle will be bicep, right? But there is also other muscle over here that work together with bicep. Is called brachialis. Okay, so because bicep is not in the choice, then the best choice will be bicep. But there is actually also, brachialis. But there are two muscles that actually perform flexions in the elbow joint, which is bicep and brachialis. Okay, so look at this bicep muscle. There is another muscle here. It's called the brachialis muscle. Both of them flex the arms in the elbow joint. So the answer will be brachialis in this question. In the shoulder elbow movement lab, this muscle is prime mover for forearm extension. Now this is extension, which is opposite of flexion. Okay, so to perform this extension, this muscle on the back over here that has to be contracted. Okay, so this muscle on the back is the tricep. So for extensions, tricep will be the muscle that will be contracted. Okay, so this is the prime mover for extensions in the elbow joint. Okay, so the answer is tricep. Now, look at this one. Okay, in the shoulder elbow movement lab, this muscle is the antagonist for forearm extension. So antagonist means opposite of extension. Okay, so this muscle will be the one that perform flexion. Okay, so when it is say antagonist of extension, it means that this muscle perform flexion. So which one's going to be? It should be easy because we already have the answer before, which is the brachialis, or if you have bicep in the choice, then you also choose the biceps. Okay, so that brachials. Okay, look at this. In shoulder elbow movement labs, okay, this is lab experiment. This muscle is the antagonist of flexion. So antagonist of flexion, okay, will be what? Will be extensions. Okay, this means that the muscle that perform extensions in the elbow joints will be the tricep. Okay, this is the muscle that is on the back that extend our arms in the elbow joint. Okay, so tricep brachii. In the shoulder elbow movement lab, this muscle is prime mover for abductions of the arm at the shoulder joint. So this is now the same lab experiment, but it is on the shoulder joint. Okay. So abduction means when we move our arms away from our body. That's what we call that abduction. Okay, just remember when someone abduct the children, it means that take the children away. Okay, so abductions is away from our body. Of course, the muscle that perform this abduction will be muscle here on the shoulder. This will be contracted. Okay, so what is the name of muscles on your shoulder? It's going to be the deltoid muscle. Okay, so this is abductions away from our body. The opposite of abduction is adduction. So adduction is when you move your arms closer to your body. So this is adduction. So the question is talking about abduction. Going to be the muscle in the shoulder over here. It's called a deltoid muscle. Okay, so this, when it is contract, it will take your arm away from your body. Okay, it is abduction. So deltoid muscle should be the answer. Now this is opposite in the shoulder elbow movement lab. This muscle is the prime mover for adduction. Okay, so adduction means when you put your arm close to your body. Okay, so like this one over here. So this is adduction. So the muscle that contracts should be muscle here. Okay, on your chest. So the chest muscle should be contracted in order to perform adduction. So what do you call that muscle on the chest? It's called a pec muscle or pectoralis major muscle. So this muscle over here, okay, the pectoralis major, will be the one that adduct our arm. Okay, so read this carefully. In the shoulder elbow movement lab, this muscle is the antagonist of adduction. So just remember, antagonist of adduction will be abduction. Okay, so which muscle actually perform abduction? Of course, this is muscles in the shoulder, okay, which is called the deltoid muscle, abduction. Okay, so remember the clue over here, antagonist of adduction will be abduction. So muscle that perform abduction will be the deltoid muscle. Okay, the last one. So read again carefully. In the shoulder elbow movement lab, this muscle is the antagonist of abduction. So what does that mean? This muscle actually perform adduction. Okay, so which one perform adduction or taking your arm closer to your body will be the muscles on the chest. It's called the pectoralis major or pec muscle. That should be the answer. I think that's all for this lab experiment or lab exercise number 2. Good luck with your lab exam. See you again sometime. Goodbye.